right, hello everybody. Welcome to another Q&A with me, Dr. Wilson. How y'all doing? All right, so a couple things to talk about today before we get started with answering questions. Yeah, I got a haircut, I know. My hair is shorter now. If I pick it, it'll still be a little afro puff. But like the Chilean miners, my hair will rise again. So don't worry, the afro will be back. But for now, my hair's short, and I'm okay with that. It was just too long. It's too hot. It'll be back. Anyway, so what's going on in conspiracy world? People, I don't know if there's any other way to describe it now, but people are just being stupid. There was some buzz online about this idea that the FDA said it was okay to treat COVID with ivermectin. No, they never said that. What actually happened was that the FDA was sued by a Mary Taylor Bowden, one of those, whatever her name is, and they just basically said that they can't stop you from prescribing ivermectin, but they're not recommending it either. It's not recommended as a use against COVID because it doesn't work. Ivermectin never worked for COVID, and it still does not work against COVID. It's a great anti-parasitic drug, but it does not work against COVID. So what exactly happened with this case? So this couple sued a pharmacy because they wouldn't fill her prescription for ivermectin, and the judge basically threw it out. It was ruled that they had no precedent to sue, that the pharmacy had every right to refuse to fill this prescription, and that's pretty much it. That's all there is to it. They didn't fill the prescription, and people got big mad about it, and they sued. And now there's misinformation about it because the FDA said, well, we can't personally stop you from acquiring and treating yourself with ivermectin. However, we don't recommend it. And there's good reason for that. Anyway, ivermectin still doesn't work against COVID. It's been tested every which way. Early in the disease course, late in the disease course, at varying doses. It just doesn't work. We have the randomized controlled trials to show that. Anyway, if you're just coming in, I'm just chatting about ivermectin. It doesn't work against COVID, despite what some people seem to be saying about it. All right, so before I get to questions again, I just would like to bring your attention to this paper, Quantifying the Impact of SARS-CoV-2 Temporal Vaccination Trends and Disparities on Disease Control. This is a really cool paper. I heard that TWIV covered it, and I haven't listened to that episode yet, but I took a look at this paper, and it was really good. Yep, I did get a haircut. The long fro is now a tiny fro, which is currently not picked out. Anyway, the gist of this paper is that when it comes to rolling out a vaccine during a pandemic like COVID, what really mattered in saving lives was the rate at which the vaccine was rolled out. Countries that rolled the vaccine out faster saved more lives. In fact, countries that rolled the vaccine out slower but still reached the same point of vaccination rates that, say, a country that rolled the vaccine out fast did, actually didn't save as many lives. And that's all really interesting because it gives us actionable items for the next pandemic, right? So it tells us that in the next pandemic, we need to roll a vaccine out as quickly as possible to as many people as possible. And countries shouldn't be stockpiling vaccines when there's vaccine inequity going on. Countries that are poor should have equal access to the vaccines as rich countries. This paper actually shows that when it came to vaccine rollout, regardless of the country's GDP, as long as the country rolled the vaccine out as quickly as possible, they were able to save as many lives as possible. So that was really cool to see as well. So the figures in this paper aren't very intuitive, so I'm not going to go through them in detail. I suggest you can read the paper, or better yet, listen to the TWIV episode about it. They probably do a good job covering it. So, yeah, really interesting lessons taught by this paper. So I just wanted to mention that here and recommend it to you all. I'll link it in the chat so that you all can have it. All right. Okay, so before I start taking questions from the chat, I had asked my patrons if they have any questions for me, and so I'm going to answer their question first. So one of my patrons, Susan, asks, I'd be curious as to how worried you are about the prospect of having an anti-vaxxer and someone with a vendetta against science, it seems, as the Democratic presidential candidate. And the second part is, what do you think needs to be done beyond what's happening now to prevent this from happening? So they're asking me, obviously, about RFK Jr. I'm not too worried about him becoming the Democratic presidential candidate. I don't think that he'll ever be nominated as the actual candidate. But I am worried that him getting this much media attention and going on all these podcasts will not be good. It's worrying that his ideas are getting such widespread attention and that they're getting praised even by the host of the most listened to podcast in the world, Joe Rogan. It's just insane. It's absolutely insane. So I do worry about the damage that will cause. We're not far away from a world where measles and other vaccine preventable diseases come back to a point where, they're relatively more common than they are now, which is non-existent. And any amount of death caused by vaccine preventable diseases is a real shame. And second, what can be done beyond what's happening now to prevent this from happening? I think it really just comes down to science education and scientists engaging with this misinformation in a way that actually debunks it. The vast majority of the scientific community could easily debunk this stuff on any given day, but they don't have the microphone, they don't have the platform, they don't have the time or really the care necessarily to take time out of their day to debunk this stuff. So, it just needs more scientists putting in the effort, even if it's just a little bit of effort, and we need better science education. Those are my answers to it, but it's a complex problem, and I don't have all the answers. Anyway, thank you, Susan, for the question. Now let's take your questions. All right, let's see what we have going on in the chat. So, Ayler asks, I'm curious about mRNA-based vaccines and their accuracy in spike protein production. How can we be certain that the ribosomes print the spikes with 100% accuracy? I've come across the proline mutation used for protein stability, yet I'm concerned about potential consequences if even a small 5% error occurs during printing. Just a hypothetical figure. Does that matter? Yeah. So I've gotten this question before, and I saw your question before I started the stream, so I pulled up some papers. So yeah, the answer to your question is yes. We can answer whether or not the mRNA in the COVID vaccine leads to an accurate spike protein when the ribosome actually reads it. And So one way to tell is just to look at the structure of what the mRNA produces. And people have done that using cryo-electron microscopy. So here is um, an entry on the protein database, the PDB, of a high-resolution structure, three-dimensional structure of the spike protein made from the mRNA used in the vaccines. It's from the paper BNT 162b2 vaccines protect rhesus macaques from SARS-CoV-2. This was an animal study that also just determined the structure of the protein product. And it's what you would expect. You could look at it, you can download this and look at it in 3 dimensions and turn it around and manipulate it and whatnot with the proper software, but it looks exactly like what we would expect a spike protein to look like. We also have this paper, N1-methyl pseudouridine found within COVID-19 mRNA vaccines produces faithful protein products. Title is pretty self-explanatory. And the key figure here is I believe, figure 5. It's figure 4. So they do a lot of Western blots elsewhere in the paper, basically showing that the spike protein is the right size as what we would expect when it comes from the mRNA used in the vaccine. And then they also use mass spec techniques, which tells you essentially what the sequence of the protein is, what the amino acid sequence of the protein is. And as the figure 4 title says, the presence of M1 pseudouridine in mRNA does not increase amino acid misincorporation frequency during translation in human cells. So there you go. The ribosome is pretty good at doing what it does, and even having pseudouridine in there, it still does its job. So, moving on, let's see. Mr. Alien 911 says, from what I've read/seen, myocarditis seems to be very disproportionately affecting young/adolescent males with the RNA vaccines. Is this also the case with the virus? So, when it comes to adolescents and COVID, so the question to ask here is, are the vaccines causing more harm in any age group than the virus? And the answer is no. I don't know the specific answer to your question. Does COVID disproportionately affect young adolescent males as opposed to adolescent females or younger males? I don't actually know the answer to that, but the question to ask in this context, in my opinion, is the vaccine causing more harm than the virus? And the answer is no. Not only does the virus cause more myocarditis in pretty much any age group than the vaccine, but it also causes other negative effects. You have to factor all of that into a risk-benefit analysis. So that's the context there. All right, Wendell Jenner asks, why did the FDA not want the data released for 75 years? Is that common? That didn't happen. So I did a video about this. Russell Brand, it was, I think, my first video about Russell Brand, where I talk about this claim that the FDA wanted to wait 75 years. No. So what happened was there was a Freedom of Information Act to the FDA asking them to release all of the files for the COVID vaccine clinical trials from Pfizer. And there are hundreds and hundreds, if not thousands and thousands of pages of documents that were submitted to the FDA for those clinical trials. And so in a Freedom of Information Act, if the FDA were to release all of them, they have to review, redact, and approve the documents for release, because a lot of those documents might have confidential patient information, which they can't just release to the public due to HIPAA laws. And it might also have proprietary information from Pfizer that obviously wouldn't be kosher to release to the public. So they have to use their clerk offices to manually read through and redact the relevant information and then have those redacted documents approved by lawyers before releasing it. And if FDA alone did that, then they essentially said it would take them 75 years. But Pfizer stepped in to help FDA complete the Freedom of Information Act request much quicker, and the documents have been coming out a lot faster. And we've practically learned nothing from them because the data was already available to us. The data is already available to us in a publicly available publication that was published in the New England Journal of Medicine on December 31st, 2020, describing the phase 3 clinical trial for the Pfizer vaccine. All of the information outlined in the main body of the paper and the supplementary materials, including the protocol, pretty much all of that is fleshed out, and you're not really learning much more by looking at the actual documents that were submitted to the FDA. All the information that they contain is distilled and communicated here in this paper. So, no, the idea that the FDA wanted to hide data, or Pfizer wanted to hide data for 75 years, is just completely false. All right, so I'm moving down the chat, so I'm not at the bottom of the chat right now, so I'll catch up, looking for more questions. Compliments on the haircut, thank you. If you weren't here at the beginning, yeah, I got my haircut. It was just way too long. Way too long. But actually, the secret is that every time I debunk someone, their soul is absorbed into my afro, and the afro was just getting too powerful, and I had to cut it. Let's see. Oh, this person says, I was happy that Lex shouted you out, although it would have been better to not have him on, him being RFK Jr. Yeah, I haven't listened to Lex Friedman's interview with RFK Jr. just because I don't want to listen to more RFK Jr. content right now. He says the same thing in every freaking interview. But I did hear that he mentioned me by name, which I appreciate. He actually did reach out to me, which I was surprised. He reached out to me asking what he should ask RFK Jr., and I did my best to give him challenging questions. But obviously, I hope he did good, but Lex is not an expert, and I wouldn't be surprised if he could not challenge RFK Jr. in the moment as RFK did his weaselly lawyer wriggling out of challenging questions. Anyway, I wish that people like Lex just wouldn't have him on if they're not ready to challenge him, because it just doesn't do enough good. Okay, RS asks, how long can you be infectious for when positive with COVID? CDC says average of 10 days, but in practical sense, can you leave isolation after 10 days and a neg rapid antigen test? This was a contentious and long saga with the CDC and how long you should isolate. So as far as I know, there are no good data on just how long after you test positive you are actually infectious. There might be good data on it, but I'm not aware of it offhand, so I can't say one way or the other. I think that 10 days after a positive test and you're testing negative by rapid antigen test, you're probably good. But, it's really hard to make guidelines for this stuff that are concrete and evidence-based and work across the board. So, yeah, I think 10 days is probably too long, but CDC sets that as a rule, and I I don't know if there's information and data out there to say that we can do better than that. But yeah, I wish we had those data. I wish people did more infectious virus studies because I haven't seen many. Rooksby asks, how do you think the efforts to prioritize vaccines to targeted high-risk groups first should be viewed in light of this paper? Does that slow the process enough to matter? So I think right. So this person's asking about this paper I just mentioned earlier, which basically says the faster you roll vaccines out during a pandemic, the more lives you save. And I think Rooksby, what we can learn from this paper is we can look at the countries who rolled the vaccines out the fastest and thus prevented the most deaths, and look at exactly how they did it, like how they prioritized who gets the vaccine first, how their public health departments distributed the vaccines and promoted the vaccines and made them available to different groups who might not otherwise be able to have access to them. I think all of those things are the important things we can learn from this paper. But I think that in any pandemic, you're going to have to prioritize people, as long as, unless you somehow have enough vaccines to vaccinate everybody, like from month one of the vaccine rollout, like you're going to have to prioritize realistically. But yeah, I think looking at the specific strategies of the most successful countries discussed in this paper is a good lesson. All right, let's see. Midas Acquisitions asks, are viruses alive? Why won't scientists answer this question? They're technically not alive. Main reason being they don't have their own metabolism. That's one of the general rules of life. It has to have metabolism, and viruses don't have their own metabolism. They kind of just sit there in space until they come into contact with a host cell. Then they enter that host cell, and then their pieces kind of start hijacking the cell to make more of itself. It can't make more of itself without a host cell. So by that definition, it's not alive, but like philosophically, the line between what is life and is not life, it, I guess, is blurred by viruses. But technically, if we want to use those kinds of definitions, viruses are not alive. Okay, Midas Acquisitions. I think we see. Okay. Well, you ask, how did you get the sequence when China claimed to not have isolated the virus on NBC back in January 23rd, 2021? Oh, you're talking about the time they failed to isolate it. They said they failed to isolate it from the market. Yeah, that's what you're thinking of. They did not isolate live virus from the market. That's all. They still isolated it from people. Common mistake that I see. So Simon Gordon asks, for the myocarditis, it was mostly the second shot. Did they confirm that increasing the spacing from 3 weeks to more reduced the risk? I remember first shot and booster less of an issue. Yes, actually. So this was figured out pretty early on. There were several myocarditis studies in China looking at males who had gotten myocarditis. One of the findings was that pretty much all of them recovered uneventfully without the need for treatment. Another result of these findings was that, if you space out the dose between dose 1 and dose 2, if you space it out like to 8-ish weeks, your risk of myocarditis goes down. And it's also good because it's more ideal for your immune system to have a longer spacing between that first dose, which is which could be considered a prime for your immune system, and that second dose, which could be considered a boost. So spacing out that time between that first prime and that boost gives you a more robust immune response. So win-win. So yeah, if you're worried about myocarditis, spacing out your doses can really help in reducing that risk. Spoonful of Dreams asks, can you tell me anything about Prof. Robert Clancy? Campbell interviewed him, but I can't find anything debunking him apart from Newcastle University saying he wasn't an expert. So I don't know much about Professor Robert Clancy, but I know that Dr. Susan Oliver over on her YouTube channel has addressed the videos where Campbell features Robert Clancy. So go check those out if you want to learn more, because her videos can teach you more than I can here. All right. Ron McKinnon asks, are there any studies that compare the severity of a vaccine reaction with that of COVID in a person? So someone who reacted strongly to the vaccine would be more susceptible to COVID and vice versa. I'm not aware of any studies that try to answer that specific question, but there is kind of a general idea that, you know, if you had a bad reaction to the vaccine, you probably wouldn't have had a great reaction to COVID either, because if you have a bad reaction to the vaccine, unless it's anaphylaxis, then, you know, it's pretty much your immune system reacting to the antigen and you suffering side effects from that. And so you could imagine if you had a lot of the antigen around because there's a virus replicating in you, that also wouldn't be good. But no, sorry, I'm not aware of any studies that specifically ask that question. All right, where are we at? We're at Midas Acquisition again. He asks, they ask, how is the risk of myocarditis less risky than COVID? What do you mean? Because, well, first of all, COVID can cause myocarditis and lots of other things. COVID can land you in a hospital, put you on a ventilator, cause pneumonia, cause mass inflammation in your lung tissue. It can cause death. It can cause actual heart attacks. It can cause blood clots and strokes. Those are all the things that COVID does. But mRNA vaccines, their most severe side effect is just myocarditis, which in the vast majority of cases resolves on its own. In very rare, extremely rare cases, it can be much more serious, but those are so rare that they don't show up in epidemiological studies. Midas Acquisition again asks, why have the UK listed COVID-19 as not HCID since March 19th, 2020? I've heard about this before. High consequence infectious disease. So I don't know about the specific guidance that made the UK determine that it's not a high consequence infectious disease. But, dude, it's killed like a confirmed 6 million people worldwide. And excess death estimates puts that number much closer to like 20 million. So, yeah. The UK might have decided to change that. Let's see if you got that date right. Status of COVID-19. Let's just see what the UK says about this. This is the this is the UK HSA website. So let's see if they answer your question for you. So they're basically saying that there's more public awareness. There's more resources to deal with it. Okay. I mean, I don't know what this HCID, what this classification really means for the UK. I mean, the viruses that are still included in this are ones with much higher mortality rates. I mean, Ebola, Marburg, Nipah, MERS, avian influenza, these are all much higher death rates than COVID. So, I don't know. But it's killed 6 million people confirmed. Almost 7 million people, I think. It's a weird question to ask. All right, I'm sure you've asked more, Midas acquisition, but let's move on. Classic Basement asks, how would you recommend talking to family members who are anti-vax? My recommendation for this would be to, number one, listen to them. Try to get a sense of where they're coming from, why their concerns, why they have concerns. And try to take an approach of like setting it up as a long conversation. Like, for example, if they send you an article that says SARS-CoV-2, if they send you an article or tell you a claim of China never sequenced or isolated SARS-CoV-2, say, okay, that's an interesting claim. I'll read whatever you sent me. How about, can you read what I've sent you here? And you can find a source and send that to them, and then ask them if you two can talk about it at some point. And just try to ask them questions to get them to think through their own logic. And then continue the conversation on a case-by-case basis. It takes work, and it's not easy, especially when it's family, but that's how I would recommend starting to go about talking about it. Brian Max says, I think you and Dr. Susan Oliver should get together and discuss politicians here in Australia and in the USA about their anti-vaccine stance. Yeah, I'm going to have Susan Oliver as a guest on soon. I just need to reach out to her. The Afro conspiracy will be a thing. Yeah, I know. Midas Acquisition again asks, how did China only lose 5,272 people to COVID in 3 years with the largest population on Earth, blah, blah, blah? Because their numbers weren't accurate. They weren't accurately reporting numbers to databases that were listing them. I don't think many people take serious stock in the quote-unquote official numbers that China reported. And we know that when Omicron hit Hong Kong, for example, and most of the people over age 60 were not vaccinated, then it caused a lot of death. So, yeah, China certainly lost more people than what they officially report. Okay, Eric McRae asks, hello, Dr. Wilson. I was wondering if the journalist Laurie Spencer has ever contacted you to discuss over vaccine effectiveness. A while back, I asked her if she is willing to talk to someone like me, I guess. That name doesn't ring a bell. I don't think so. Wendell Jenner asked, would you go on Joe Rogan to debate RFK if asked? I mean, maybe. That's never going to happen because I'm a practical nobody, and the only reason RFK Jr. and Joe Rogan want someone to come on and debate is if it's someone like Peter Hotez, who has long been a infectious disease researcher and a voice for the vaccine community, and or for a voice for vaccines. So, he's not going to have on someone random. They want clicks, they want views, and a debate between Hotez and RFK Jr. is what's going to bring the clicks. Anyway, to answer the question, maybe, probably would be my answer. It's one of those situations where I'd have to be in it to know what I'd actually do. But, yeah. Maybe, probably. Let's see. Next question. Covert White Rabbit asks, are there any easy or successful ways to explain to internet sleuths the difference between research and confirmation bias? Yeah, the way I would describe it is I would ask them, were you trying to disprove your belief? Because that's what honest research and what the scientific process actually does. The whole approach of the scientific method is to try as hard as you can to disprove your hypothesis. And if you cannot disprove your hypothesis through the experiments you've set up, then you have to accept your hypothesis. So, yeah, I don't know. That probably won't get to them, but that's how I would put it. Did they actually try to disprove their belief? And if they didn't, then they're just confirming their bias. Okay, next question. Let's see. Midas Acquisition again asks, what's your perspective on WHO Director Tedros refusing to get vaccinated? I highly doubt that. Why would you think that he's not vaccinated? Yeah, this is what comes up. WHO chief is vaccinated against COVID, contrary to false claim. We're not going to go through the article, but I mean, I would find it ridiculous if he was actually unvaccinated. So there you go. Let's see. Harry Kirk asks, is it actually possible to determine the origin of a virus in all cases? It seems on Twiv they have said it's somewhat rare to make a solid determination as to origins. So, I guess if you're asking who was the patient zero and where did they get it from, identifying that, that's usually really hard. Because, excuse me, usually by the time you notice that there's a new virus that is spilled over to humans, the patient zero and the animal that they got it from are gone. In the case of SARS-1, you can, in the case of SARS-1 where they didn't close the market and call all the animals right away, you can go to the market and see, oh, this virus is in these animals and it's circulating. This is probably where it came from. But as far as where, what was the first animal to pass to humans and where did that animal get it, that all is much harder to figure out. Midas Acquisition again says, how do you explain China losing only... Okay, never mind. That's the same question. I already answered that. All right, starting to catch up to the end of the chat here. Oh yeah, I just pulled up the same AP News article. I'm catching up. Thanks for posting that, Les. Let's see. I appreciate all the well wishes and the thank yous. I do see those in the chat. Thank you very much. Mike Frame asks, new article I skimmed about COVID-19 treatment using siRNA. Do you think this could be effective and affordable? I haven't heard about that. That'd be cool. Oh, why did I just type that in? Potential of siRNA and COVID-19 therapies published in Frontiers. That's from 2023. There's also a 2021 paper in Cell, a SARS-CoV-2 targeted siRNA nanoparticle therapy for COVID-19. Yeah, I mean, it would be cool, and I don't know all the details about it, and I'm not gonna read about them right now, but just seeing that there was a paper in Cell from 2021 and now a new paper in 2023 makes me think that there were some problems with it. I mean, more treatments is always good, so hopefully it does have potential and it ends up working out. But affordable, I don't know. I don't know. I don't know how expensive siRNA drugs usually are. Affordable in America? I doubt it. Affordable other places? Maybe. But that's just me spitballing. I don't know. All right. South Canadian asks, do you have close family that speaks nonsense science? I have a PhD in chemistry with a target in drug design and still hear craziness about pharmaceuticals. Yeah, I have one family member who thinks that COVID was a lab leak, and that's been frustrating talking to them. So, yeah, I feel like every family has those kind of people, at least one. There are others in my family who have, I guess, I don't really have any anti-vaccine family members. But yeah, it's tough because when family does that, it's just like it can drive a wedge between you. And that's not fun. But sometimes, it's just not worth having the conversation if it's going to like ruin your relationship. So that's kind of where I am with that one family member. We just don't talk about it. Subtle Salmon asks, have you taken a close look at the paper RFK was talking about with COVID affecting Jews and Chinese people less? I would love a deep dive debunk on how he was misrepresenting it. Yeah, I did look at the paper. This was weeks ago when that clip of him was going viral. But from what I remember, the paper was looking at SNPs, single nucleotide polymorphisms. I think that's right. It might have been more broad mutations, but we'll go with that for now. They were essentially looking at mutations that were more clustered in certain ethnic groups or races that may or may not contribute to a more severe outcome of COVID. However, the paper was basically just theoretical. They didn't link anything they found to actual clinical outcomes, and so the literature as a whole doesn't really support the idea that any one particular ethnic group or race is more prone to severe COVID. The only reason we see that, for example, in America, Hispanics and Black people are dying at higher rates of COVID than their white counterparts is that more often Hispanic and Black communities are underprivileged, they're more impoverished, they have less access to good health care, they're already less unhealthy due to things like food deserts and, just general poverty, making them, more often have poorer diets, and again, less access to health care. So those are the reasons, not just the fact that they are Black or Hispanic, but all of those reasons is why they're more affected. But yeah, I'll consider making like a full video about it. RS asks, is it possible to test positive on a rapid antigen test and not be infectious? I.e., I have a friend who is positive on a rapid 11 days after initial day. They are wondering if they can leave isolation. Yeah, it's definitely possible to test positive for COVID on a rapid antigen test or a PCR test and not be infectious. In epidemiology, there's something called an 80-20 rule, which is that 80%, it says that 80% of the cases come from 20% of the people infected. I don't know if that rule is like universally true, but it's something that I've seen floated around in epidemiology circles. Maybe someone will tell me I'm totally wrong there, but the point is, not everybody who gets infected is going to shed infectious virus, or at least not enough to infect another person. And there are multiple factors that affect that. The only way to know if someone is actually shedding infectious virus is to take samples from them, whether it's their throat, their nasopharynx, or just their material from their coughs and sneezes, and do an infectious titer on it. And the problem with that is by the time you've done an infectious titer experiment, which takes up to like a week or longer to do, it's too late. It doesn't matter. They've already either been in isolation for so long that they're probably not infectious anymore, or they've already infected other people. So it's not a feasible way to do public health. The safest way is to just, if you test positive, then take those precautions. But in the case of your friend who is 11 days after the initial day and they're still having that rapid antigen test, they're probably fine. They're probably not infectious. Just, wear a mask if you want to be safe. But you're past the 10-day CDC recommendation, so you're good either way. 502 Conversations asks, will you debunk the trigonometry episode, the truth about vaccine injuries? Yeah, actually. I've been meaning to put that on my list. So I haven't checked that out yet, but I will do that now, and that'll be one of the videos I make next. RS again asks, why aren't there more trials on a longer duration of Paxlovid, given that there is a proportion of those who get rebound illness? Okay, so this is a common thing that bears repeating. Paxlovid is an antiviral. The way an antiviral should work, the way it does work, is it works early in the infection. So while the virus is still ramping up, while it's still replicating, that's when you want to take an antiviral. That's when the antiviral is going to work to stop that replication and stop you from getting to that point where the virus reaches that critical mass where you're then at risk of progressing to more severe illness. Past that phase, an antiviral is not going to do much for you. And the rebound is not caused by the virus replicating more, or it's not caused by Paxlovid. The rebound, the so-called rebound, is just the normal course of COVID, where in those first few weeks you feel crummy, you feel sick, like general flu-like symptoms. That's the virus replicating. And then later you get you might start to feel better, and then later you get this big immune response, which is the inflammatory phase of the disease. At that phase, doctors will typically try to treat you with things like corticosteroids or tocilizumab or baricitinib, things that are going to dampen that immune response and get it under control so that it stops causing damage to your body. Because that's what can happen with the immune system. It can overreact, cause a lot of inflammation, and cause damage. All right, moving on. John Horak. Oh, wait, there's a super chat. 6 for $10. Thank you. Thank you for that. You don't have to do that, but I always appreciate it. Yeah. Thank you very much for that. It's okay. Sorry, John Horak. Let's get back to your question. How can you explain all 100 PubMed abstracts with fancy words about spike proteins and ivermectin? Pretty easy. You can just look in the abstract and ask, is this a randomized controlled trial? If the answer is no, then you can just kind of dismiss it. Because, like, here's how it works. If you want to build a case, a scientific case for something like ivermectin working for something like COVID, you want to first say, okay, does it work? Is there a theoretical mechanism by which it can work? Okay, let's talk about that. Let's write that out and put it out there. Okay, next step. Let's see if it works in a petri dish. Let's see if it can kill the virus or stop it from replicating, whatever. Okay, cool. Next step. Let's see, maybe does it work in animals? Maybe there's not a suitable animal model, whatever. Let's go to small scale, like observational trials and see whether it works. Okay, great. Let's keep moving up. Ultimate test for a drug like ivermectin to see if it's effective against COVID would be to do randomized controlled trials. We've done that. There are several randomized controlled trials against ivermectin. They all show that it doesn't work. So yeah, if someone sends you a PubMed abstract saying ivermectin, spike protein, blah blah blah, just look for whether or not it's a randomized controlled trial and say, sorry, we have randomized controlled trials that show it doesn't work. All right, Sally Bruckner asks, lab leak advocates seem invested in attacking Fauci, but seems to me what's important is, are virology labs sufficiently regulated? Do you think they are? Yeah, I think that they are. I mean, I frequently work in a BSL-2 plus lab. So BSL-2 plus is kind of a middle ground between BSL-2 and BSL-3. And, we don't work with anything that's super dangerous in there. And the rules are still pretty strict. There has to be negative pressure. There has to be an antechamber between between the lab and the hallway. You need to wear disposable lab coats. You need to double glove, wear safety glasses. You can't bring any of your personal items in there, all that stuff. And BSL-3 and BSL-4, are much, much higher than that. And there are lots of boards that oversee grants that would fund research with viruses. And scientists aren't dumb. I mean, we're human, we make mistakes, but we're not dumb. There's not a lot of room for catastrophes to happen when it comes to virology labs. Not saying it's impossible, but it's pretty well regulated, I would say. All right, moving on. Okay, Zeddy asks, have you noticed any examples of a content creator having the opposite trajectory of someone like John Campbell, where someone was misleading followers and has now seen the light? Interesting. I don't know of anyone during the pandemic who fits that profile. I think there's a guy, Brent Lee. When did he leave his conspiracy views behind? I don't quite know. But his name's Brent Lee. He used to be a conspiracy theorist, and now he runs a podcast. Some dare call it conspiracy. It's really good. I've listened to a few episodes of it, and I enjoy it. Yeah, he co-hosts it with other people, I think. But yeah, I can't think of anyone who, during the pandemic, started off spreading misinformation and then switched. All right. Oh, okay. Ayler is asking a follow-up question. You're just asking the same question. You say, I'm curious about mRNA-based vaccines and their accuracy in spike protein production. I did answer this before. In case you didn't see it, we have cryo-EM 3D structures of the spike protein produced from mRNA vaccines. It looks as we would expect, and we have papers like this that examined whether or not the sequence is accurate, and figure 4 here, the title says it all. The pseudouridine in the mRNA does not increase amino acid misincorporation frequency during translation in human cells. So Ayler, if you didn't see that before, there it is again. I hope that helps. All right, moving on. Marco Mont, I'm not sure if this is a question for me. You asked, did you see what was first called BA6 is now a variant of concern by WHO and named BA2.86, I believe. It's in Michigan. Yeah, not surprised. SARS-CoV-2 is a virus. It's going to keep mutating. We're going to keep seeing variants. Until a variant is causing the same rates of severe disease in vaccinated people versus unvaccinated people, I wouldn't be too worried. Vaccines and antivirals are holding up pretty well. Kaylin B asks, is myocarditis particularly a side effect of the COVID vaccine, or is it all vaccines, we just don't hear about it for other vaccines? What does myocarditis have to do with the COVID vaccines? So, yes and no. Smallpox vaccines were known to cause myocarditis back in the day, but pretty much most other vaccines do not cause myocarditis. What does myocarditis have to do with the COVID vaccine? We don't know exactly how COVID or the vaccine cause myocarditis, but there are hypotheses with varying amounts of evidence. The short explanation is that people think the data points to it being a phenomenon called immune cell infiltration, where immune cells kind of infiltrate the heart tissue and cause inflammation there. We don't know exactly why that happens, though. So yeah, I would expect that we're going to know more about that in the coming years. Joey LaFronde asks, "Hey, Dr. Wilson, is John Campbell a paid shill?" I mean, he makes a lot of money making his videos, which are total bunk. Is someone else paying him to do that? I don't think so. I haven't seen evidence of that. Probably not. He just makes a lot of money on his YouTube channel. Rooksby asks, "Dr. Wilson, maybe the recent David Quammen would be helpful for your lab leak relative. He ran down in great detail all of the LL arguments in a respectful and non-disparaging way." Yeah, so with that family member of mine who believes, or last we talked about it, believed in the lab leak idea, I mean, I would send him experts talking about it, like TWIV episodes and whatnot, papers, but it didn't seem to get him to budge, and then they were just kind of doing personal attacks, and I didn't need that from a family member. So yeah, we just haven't talked about it, but maybe one day we'll talk about it again. I don't know. David Palermo asks, "As far as I know, I've not had COVID. Is there a way to find out if I've had it?" Theoretically, if you were to test yourself for antibodies and see that you have antibodies against like the N protein in the SARS-CoV-2 genome, then that would be an indication that you have been infected or exposed to it before. But if a negative test on your N antibodies, on antibodies against the N antigen, wouldn't necessarily mean that you haven't been infected either, because maybe the antibodies are just at low levels such that you can't detect them. So there's a way to find out, but it's not like foolproof. Okay, Midas Acquisition once again asks, "Do all races share the same level of spike proteins, or are the vaccines themselves tailored for different races? A paper on PubMed states blacks have the lowest level of spike protein count." Do you know that? You know that spike protein is not made by people, right? So I'm not sure what you mean by all races share the same level of spike proteins. Like, it's a viral protein. Our cells don't make it. Yeah. Okay, moving on. Alana McCollum asks, "I'm not anti-vax, but so many young people are having blood clot issues, like Tori Kelly, the singer, in her lungs, legs, and around her heart. Marquise Godwin, NFL player, clots as well, vaccinated." Okay, I guess it's not really a question, but yeah, I mean, those things happen in a normal population. People get blood clots, and young people get blood clots too. It happens. The question is, does it happen at an increased rate in vaccinated people? No, it does not. mRNA vaccines do not cause blood clots. Myocarditis is a rare side effect from them, but not blood clots. Tom Ardens says, "I hope you donate your afro to charity." I don't think anyone wants that hair. No, but I did give my barber a good tip. It was my first haircut in 3 years. It had to happen eventually. Okay, Midas Acquisition again. If the virus spreads via respiratory droplets and fomites, why were test samples not taken from nasal pharynx? It's nasopharynx, and not a basic saliva sample. I mean, you could take it from a saliva sample, but the nasopharynx is the best place to take a sample from, because if you're in a stage of the infection that is too early to detect it in your saliva, then you're going to get a false negative. So you want to be able to get deep down in there where the virus is going to start replicating in order to get the most likely correct answer. And again, Midas Acquisition asks, in regards to China, if the virus came out of China and China have the largest sample size of a population, why wouldn't the Chinese data be most valuable, especially when it comes to autopsy? I'm not sure what you're asking here. It's not up to us whether or not the Chinese government wants to share their figures. Like, they just don't share accurate figures. But we did get a lot of information coming out of China from scientists who were doing a lot of early work to identify the virus, characterize the disease, and whatnot. I mean, a lot of good work came out of the scientists who were willing to do the work and publish it online. I'm starting to understand your questions less and less, Midas Acquisitions. Anyway, moving on. G. Ferraro asks, Dr. Wilson, what are your thoughts on this fall and all the vaccines? I am older and thinking of taking a flu, a new variant booster, and RSV. So this fall, I think most people do not need a new vaccine, a new COVID vaccine, that is. I'm going to be getting my flu shot. When is it? It's August now, so September, October-ish. Like September or early October-ish is when I'm going to get my flu shot. My son is going to be born in October, so definitely want to get it before then. But yeah, I don't think most people need a COVID vaccine. If you are older and at higher risk, then it is recommended that you get a booster, and I think that's reasonable. If we're expecting a winter surge and you want that extra protection, then it's reasonable to get a new vaccine, because as you get older, your T cells, which are what's really going to help protect you against severe disease. This is theoretical, by the way. This is not empirically demonstrated, but this is just what makes sense. Your T cells kind of become less functional, and so you have to rely more on those neutralizing antibodies to protect you against disease. And so getting a boost with a vaccine is going to replenish those numbers of antibodies and possibly give you more protection during a winter surge. And RSV, I mean, I'm really happy that RSV vaccines are coming on the market, especially with my son being born in October. You know, we didn't have anything to protect my daughter against RSV, and we had to worry about that, especially when she went to daycare. But anyway, the vaccine for children isn't a true vaccine. It's like a passive vaccination, it's a monoclonal antibody infusion. But for older individuals, there is now an RSV vaccine, and I think that's totally worth getting. Sorry, that was a little long-winded. Okay, moving on. Meister Murphy, good to see you too. The number of cell phone accounts that went dead in China is indicative of a much higher than reported death rate. I don't know anything about that, but I am certain that their death rate was much higher than what was reported. Ultimate Weapon 2000 asks, do you see any warning signs with the new variant? None yet. I don't think that there's any reason to believe that this new variant is going to be able to evade our immune memory or antivirals any more than any other variant. So yeah, I wouldn't worry about it yet. Okay. SAR, or sorry, AZ16 gave a super chat. Thank you very much. I really appreciate that. Oh, and another super chat from Anubis. Thank you. You say get on the Duran, George Calloway, Kim Iverson, or even Rogan. I don't know what I've never heard of the Duran or George Calloway, or George Galloway. I don't know if I should know who those people are. I've heard of Kim Iverson, Rogan. I don't think I'm ever going to go on Rogan. There's just, I don't think that he would ever do that. He would ever invite me. But yeah, I'm happy to go on these platforms and try to talk about the misinformation and why it's wrong. Ben asks, which politician is most scientifically competent in your opinion? I don't know. Maybe Scott Kelly, the astronaut. I honestly can't tell you much about many politicians' scientific literacy because I don't really listen to them very often. So, yeah, I would guess Scott Kelly. Tim Farrell asks, WTF are we going to do next pandemic as the anti-movement has continued to grow? How do we get truth through to the people? It's a good question, man. Yeah, I worry about that. I worry about what's going to happen during the next pandemic because there will be one eventually, because that's the way nature works. What we're going to do then, because it feels like there's already going to be a group of people who are entrenched in this idea of never again, we're not going to lock down, we're not going to mask, oh, blah, blah, blah. And I mean, the next one could be much worse than COVID. And I feel like the people will still be entrenched in those views, even in that situation. So I don't know what we're going to do, man. Just, like I said earlier, we have to improve science education on the very basic level, and we have to get, maybe the answer is to get more science influencers, like get more people who can communicate science effectively on TikTok, on Instagram or YouTube, get them on the platforms, like wherever the misinformation is. I don't know, man. There needs to be more and better science communication. All right, moving on. Let's see. Midas Acquisitions is going off in the chat. Cool. Margot Moyes says, I still hear people claiming that COVID-19 virus has been isolated. Is that different to the genetic sequence being known? I'm from Australia and believe a lab here was the first to isolate the virus. No, so the first lab to isolate the virus was in China. They published their results in the New England Journal of Medicine. They sequenced the genome, they grew it in cells, and they took a they took a negative stain electron microscopy picture of it. There was a lab in Australia that isolated the virus from their first cluster of cases. But yeah, it's definitely been isolated. G. Ferraro says, Dr. Wilson, that's a lot of vaccines. Yeah. I mean, 3 vaccines it's not a ton. If I want to go to where my dad's side of my family is from in Southern Africa, in a country called Eswatini, I have to get lots of vaccines. So your body, your immune system can handle it. Mark Mixon says, I had second booster April 22nd and tested positive late last fall. Should I get another shot? If you're not high risk, I don't think you need one. Congratulations on the upcoming birth of your son. Thank you. It's my second child, and we're very excited. Brian Mack asks, why are Dr. Fauci and Bill Gates still classed as the bad guys in all this? I don't know. I mean, the hate for Bill Gates, I don't understand, because he's just a rich guy who does a lot of like donating to vaccine related causes. Like, he's just a rich guy. Like, he's not an expert. I don't know why people treat him as like, oh, he said this and it was wrong. Like, well, yeah, because he doesn't know what he's talking about. He's not a scientist. He's just a rich guy. And Fauci, I mean, you know, he's the scapegoat. He's the boogeyman that people need to unleash their anger against. It's weird. Base Theory asks, you may have already answered this, but will you be debunking RFK Jr.'s interview with Tucker Carlson? Oh, I didn't know he did an interview with Tucker Carlson. So, see, the thing is, part of me thinks that any future RFK video I make, it's going to be the same stuff, because he literally says the same stuff in all of his videos, in all of his interviews. So I don't know if I have plans to cover another interview of his anytime soon, at least. I will be inviting someone on my channel to talk all about him and his dealings in the past. And she knows a lot more about that stuff than I do, so that should be cool. Let's see. John Wells asks, to your knowledge, is the incubation period for the latest variant still around 3 days for symptoms to show? I think that's about right. Omicron incubation period is fairly short, on the order of 3 days, and I don't think that has changed much with this variant, to my knowledge. Hope that helps. Okay, let me just address this from Midas Acquisitions. They say, what's higher, 95% or 99.99997% survival rate? I don't know what the context of this is if you're replying to someone, but COVID was never a 99.997% survival rate. Before vaccines, the case fatality rate was always around between 1 and 2%, closer to 2%. And that's fairly high for a virus that was infecting a lot of people. Post-vaccines and post-population immunity, which was very costly, it's a lot lower. Anyway, Ultimate Weapon 2000 asks, I keep seeing anti-HPV vaccine commercials. Has there been some sort of incident with those vaccines, or are those commercials fake? I'm not sure what commercials those would be. I haven't heard of any news about HPV vaccines. It's just the same old misinformation that I'm aware of that goes around about them. David Palermo asks, I've been wearing masks since early on in the pandemic, only N95s. How effective are they? Might be one reason I never got COVID as far as I know. They're pretty effective. I mean, an N95 specifically, if it has a decent seal, I mean, you're not going to get a perfect seal unless you get it fit tested, but an N95 compared to a surgical mask is going to give you a better seal around your face, which is giving you some extra personal protection. Whereas things like surgical masks, they're mostly for you to protect others, like preventing you from spewing stuff out of your face into the environment. They're source control. But yeah, N95s, they're going to give you some protection for sure. 6 asks, they don't care about science education. They care about what their favorite YouTuber slash podcaster has to say on it. Sorry. I think I saw the question mark, but I think you might be just replying to someone else. Not really a question. Anyway, I'll move on. Dayan asks, did you know about New Phillips' claims about ADN contamination? ADN contamination. New Phillips. Are you talking about? Okay. I think I know what you're talking about. DNA contamination. Right. So on Twitter, there's this guy, Philip. I forget how to pronounce his last name, but so one of the videos I made recently about supposed DNA contamination in COVID vaccines, which is all garbage, like super garbage. It's so funny how bad McKernan's little blog post was on it, and I call it a blog post even though it's a preprint because that preprint server has zero quality control, so he could literally post anything on it. But it's hilarious how bad it is. But he keeps claiming that others have replicated his findings, and this Philip guy, who's a professor at I think Stanford. That might be wrong. I forget. But he's a professor somewhere in California. He claims that he got a vaccine vial fresh from the clinic close to his lab and found DNA in it. Shocking. But has specified that he has found, he actually quantified it much better than what McKernan did. He actually had a decent standard curve and his PCR passed efficiency, and he reported CT values with decimal points and all that stuff. Just basic stuff. And he said that it's below the allowable limits of what the FDA sets. So yeah, there you go. It's below the allowable limits, which is the whole point. Because if it's below the allowable limits, it's pretty much trace amounts that aren't going to have a biological effect. I mean, 3 micrograms, I think, of mRNA vaccine did not work in children ages 5 to 11, I believe. It didn't produce enough of an immune response. That's 3 micrograms. Go 3 orders of magnitude down, and you get to these trace amounts of DNA in the vaccines. It's not going to do anything. But yeah, his latest, this Philip guy's latest concerns is that the DNA is actually going to integrate into our genomes. So he's reasonable enough to do a decent qPCR and quantify the DNA and say, yeah, it's below allowable limits. But what about integration? No, it doesn't work like that. Random bits of DNA are not going to just integrate into your genome. Anyway, I could go on about that forever, but it's ridiculous. Les asks, what do you think of the universal flu vaccine candidate? I get asked this every once in a while, and I always say I will believe it when I see it. It's, a universal flu vaccine is a tall order. That would be a big achievement. It's really tough to get one that works as a universal flu vaccine. I hope someone accomplishes it, but I'll believe it when I see it. Anubis asks, why do people think aluminum is unsafe in vax or in deodorant, etc.? Because they're chemophobic. They're afraid of chemicals, and they're afraid of metals. They don't really understand chemical context, and that dose makes the poison. They're just afraid of chemicals. Neeker Breaker asks, do you happen to know of any good resources, for scientists to improve their skills at communicating science to the public? It's a good question. Boy, I mean, so I did a lot of things in grad school to help me get better at teaching science. Um, so like I taught some lectures in my professor's course. I, volunteered on weekends to teach teach labs to like grade schoolers. I did outreach volunteer programs for with the university. So I did a lot of practice, but as far as like as far as resources specifically geared towards that, I don't have any on hand. Uh, Neeker Breaker, send me an email about that, and I'll try to look for some resources for you and get you some of them. But definitely send me an email on that. uh, let's see. Moving on. Uh, Looking for some questions. Um, Emil Palm asks, Dr. Wilson, what is your response to the people that say that Big Pharma is suppressing real cures to cancer/COVID in order to make more money? I say that if they had those cures, they would make more money. So there's a nice story. It's told in a book I have on my shelf over there. The book is called The Philadelphia Chromosome by Jessica Wapner. It tells the story of this drug called Gleevec, or imatinib, are the two names for it. It's a drug against chronic myelogenous leukemia. It's practically a cure for it. The story is just incredible, where these chronic myelogenous leukemia patients were just, their survival rate was just so low, and they would be sentenced to this really painful death. And it's terrible. But this drug Gleevec, it practically, they have to take it for pretty much the rest of their lives. People think maybe they could go off the drug, but then they risk remission or a relapse, and that study hasn't been done on whether or not a patient would actually relapse. But anyway, the point is that drug is super expensive. It's a fantastic drug, and the story behind it is so cool, with awesome scientists doing so much work to get it approved and into patients who needed it. And it's so freaking expensive that it's what the hell? It's sad. It's sad that pharma can do that, can make a drug like that so expensive. And the guy who was instrumental in making it happen, Dr. Brian Drucker, he's very upset with how expensive the drug is. The point is, if there's a cure, there's going to be a high demand for it, and pharma can charge whatever the hell they want for it, basically. So if they had cures for cancer, they would definitely sell them. All right, let's see. Moving on. All right, I'm starting to see fewer questions. Yeah, not Philip McMillan. The Philip I was talking about earlier was, it's Philip B something. I don't remember. Okay. David Palermo asks, my sister won't get the COVID vaccine because she thinks it wasn't tested enough. She got COVID. How can I convince her and others that it has been tested rigorously and testing wasn't sped up? So the way I usually explain this, and this might be a lot to explain, feel free to clip this video if you would like to send it to your sister. But here's what I would say. Most people are confused with the timeline of COVID vaccine testing. And they'll often say vaccines normally take 10 years to get to market. I usually try to ask people when they say that, what do you think those 10 years involve? Usually they don't know. And the truth is that those 10 years usually involve a lot of okay, let's do a preclinical phase and then submit to the FDA and then wait a while. Wait to hear back from the FDA. Okay, we get the green light. Cool. Let's go on to phase 1 trial. Okay, got to recruit for the phase 1 trial, got to organize it, assign sites, do all this stuff, do the trial, gather the data, submit to the FDA. And then, again, wait a little bit, wait a while until the FDA reviews that. Then you can say, if they give you the green light to go to phase 2, then so on and so forth, phase 2, phase 3. And each phase gets bigger and bigger, so it takes more and more work to set up the phases. So there's basically a lot of waiting in the FDA queue for reviews. And, sometimes there might be issues with fundraising, whether or not there's money for funding all these clinical trials because they're very expensive. So those are the two big holdups, is FDA wait queue times and money. With COVID vaccines, those weren't issues. Money wasn't an issue. Thankfully, lawmakers decided, let's put a bunch of resources into this, and FDA prioritized COVID vaccine review. So they didn't have to, when they submitted for review, that review didn't have to wait in line behind a bunch of other drugs. FDA got the data, they started reviewing it right away. So those were big differences that allowed the clinical trials for COVID vaccines to happen very quickly. And thankfully, all of the basic research, which would happen before that usual 10 years to make the actual timeline from a drug's bench to bedside journey more on the order of 30 years. And mRNA vaccines have had 30 years of basic research, basically. So a ton of research went into the technology, and the clinical trials didn't really skip steps. So there's all that. And then on top of those clinical trials, even better, what we have is phase 4 trials, which is surveillance of the 13 billion doses of vaccines that went into the population. That was all monitored by various institutions, governments, and looked at by academic labs all over the world, crossing affiliations, crossing governments. There's so much data that all says the same thing. So yeah, I would say something like that. Hopefully that helps. Okay. Where do you, Meister Murphy says, where do you think we'll stand on cancer cures in 10 years? I hope there are a lot more. I hope there are good cancer vaccines. I hope we make progress on drugs. I know a lot of companies are focusing on monoclonal antibody treatments for cancers, which is nothing new, but they're looking at, just new targets, new approaches. So I hope we've made a lot of progress on treating cancer in the past 10 years, and I hope that continues in the next 10 years. All right. I hope you guys can't hear my dog licking himself. He's like 5 feet away from me. All right. Sam Shinnick Guitar asks, "Hi, Dr. Wilson. Would you think about making a video about long COVID? I feel like not enough people are aware of the risks, even with a mild infection, and it'd help with understanding it." Yeah, I can put that on my list. I would have to do quite a bit of reading about long COVID, and maybe find like some. Actually, there's a lot of misinformation about long COVID, so that probably wouldn't be hard to find. But yes, I can keep that in mind as a video topic for sure. Tim Farrell asks, "Just curious, what would you do different, if anything, if a new pandemic emerged to engage the public and debunk misinformation?" What I would do different to engage the public and debunk misinformation if there were a new pandemic? I think I would just be more, I would do my best to be more on top of what the actual conspiracy theories were and like what misinformation was going around news cycles. Because with COVID, my channel was just barely starting when COVID was coming out. And at the time, my thoughts were that this would probably be another SARS-1 where it kind of just fizzled out. But I was wrong about that, and here we are. Yeah, I would just have to be way more conscientious about being on top of whatever misinformation is trending at the time. Anubis, another $5 super chat. Thank you very much. They ask, they say there's a convincing, quote unquote, convincing documentary called Second Opinion about Latrili as a cure for cancer. Maybe you can do a video on it. Okay, I'll keep that in mind. I haven't heard of that, but I'll keep that in mind. All right. Dayan says, "What's your opinion about the last John Campbell video? Life insurers confirm excess young deaths." I haven't seen it, but Susan Oliver made a video about it, so go check that out. John Campbell's just ridiculous. I mean, he's so lazy in all of his videos now. Six says, "I would love for you to give us a bookshelf tour. I already tried to read your titles." I could do that someday, yeah. I got like these, I think these two shelves, or yeah, like half this shelf is fantasy books, and this entire top shelf are fantasy books as well. The rest are like nonfiction books, and I've got textbooks down here and random stuff over to the right that's not organized. But yeah, I could do that someday. All right, let's see. Emil Palm says that they're a fourth-year med student and is familiar with CML and imatinib, but never read the story behind it. Yeah, I highly recommend The Philadelphia Chromosome by Jessica Wapner. It's such a good book. It's one of my favorite nonfiction science books. What is this Eminem rap in the chat? Let's see. Grego says, "Where's your hair?" Just kidding. Yeah, I did get a haircut. It was too long. It was just too long. My wife was also like waiting for me to get a haircut. More money needs to go toward cancer prevention versus cures. I can agree with that. Prevention is better than cures. However, I will say with cancer is kind of an inevitability if you were to live long enough. Like, it's kind of built into our DNA where, like, you know, we have all these mechanisms for our cells to work together in our body to make us us. And those systems that allow them to work together break down over time. And just the longer you live, the more likely they are to break down and cause cancer. That's putting it pretty simply, but yeah, we also need cures. But I totally agree. Prevention, you know, a lot of people get cancer young. And yeah, prevention is super important. Let's see, it looks like we're winding down here with questions. It's getting to be, oh my God, it's almost 11. So I'll definitely have to end soon because 11 o'clock is late for me now. Meister Murphy says, Dr. Drew grinds my gears. Yeah, me too. I've sent him several emails that he has not responded to over the past few weeks. It's crazy. Hey, somebody mentioned you on Leftist Mafia just now, and I'm subbed. I don't know Leftist Mafia. Who's Leftist Mafia? I'm sorry. Should I know who they are? I feel really bad if they're shouting me out and I don't know who they are, but I don't know who a lot of people are. So please don't hate me. I found their channel. Oh, looks like they have a video on Steven Crowder, dragging him. That's great. Steven Crowder is terrible. Awesome. Looks like a cool channel. Okay, anyway. Okay. All right, let me try to speed run these because I wanna get through your questions, but I also need to go to bed soon. Let me try to go in order here. Margot Moyes asks, there are still claims the mRNA vaccines are causing cancers. Isn't it a fact that mRNA vaccines are actually a great advance in the more targeted treatments of the future? Yeah, mRNA vaccines for cancer treatment is a hot area of research right now. COVID mRNA vaccines, yeah, people claim they cause cancer. It's total garbage. They do not cause cancer. There is no even conceivable mechanism by which they could cause cancer. I know that a certain Dr. Peter McCullough says otherwise, but his paper claiming that was so bad, and I did make a video on it. So yeah, I think I described it as basically like it's a bunch of word salad where basically he's saying if my light in my bedroom and my stove downstairs both run on electricity, that means if I switch on the light for my bedroom light, that also affects my oven because they both run on electricity. No, that's not the way it works in biology. Just because cancer can be affected by a certain gene cascade, that doesn't mean that affecting it's going to cause cancer. Like, it just doesn't work like that. Anyway, I said I should speed run these, but I'm rambling again. You should get together with the Unbiased Science podcast people and do an interview. I love Unbiased Science. I do know who they are, and I know their podcasts. They actually, when I made my Robert Malone on Rogan video a while back, they did reach out to me, but that didn't. I think I responded, but then they didn't respond back. So that's all good. No shade on them, but yeah, that would be cool to work with them. So many scientists love fantasy fiction. Maybe sometime you could do a video on your favorite books and authors when you read for fun. Dude, I haven't read a ton of fantasy series, but I have ones that I love a lot, and I could talk forever about them. But I don't know. Is it like self-indulgent to be like, I'm going to talk about my fantasy books I like on my YouTube science channel? Like, I don't know. I could talk forever about Malazan Book of the Fallen series, but anyway, moving on. Okay, Shouts asks, hi Dr. Wilson, not sure how much you know about PCR. I keep hearing that most qPCR assays require that samples be ran at a minimum of 40 cycles. Why is that the magic number? Why not 30 or 50 or 100? So it depends on the lab. Like the different clinical laboratories will test. They have different platforms, different methods, different like qualified criteria for their COVID PCRs. Excuse me. So some labs will run at 35, some labs will run at 40, some labs run at 38, 42. The point to remember is, let me just bring this up so I can make the point more clearly. The important thing to remember about qPCR tests, where is this? Is that it's done in real time, and it's measured in real time as well. So this is just an example of, like, this is a blog by a virologist just talking about qPCR and COVID PCR, and this is an example data set from him where you have all of these lines, which are negatives, either negative controls or negative samples, and you have this one positive control, and this is run for 55 cycles, and the negatives are still negative, and the positive came up around, looks like cycle 30. So it will register positive at cycle 30. So it's not like you run for 55 cycles and whatever's positive at the end, oh, that's positive. No, you get like a quantifiable amount in your qPCR based on what cycle it passed the threshold. Okay, I really got to get through the rest of these questions. I want to answer them all, but N64US says some say phase 4 monitoring is too passive, leaving it to the patient/doctor to report, and phase 3 was active monitoring. Is there a chance a lot of COVID vaccine side effects have been missed? So this is a myth that phase 3 is better for detecting adverse events. Phase 4 is better. The reason being that phase 3 trial is going to have like around 40,000 people divided into at least two groups. If you have, so when you finish that, you're going to know that there probably aren't any severe adverse effects that are like common. But you're not going to know if there's an adverse event that's like 1 in 100,000, or you're not likely to see that. You're not going to know if there's a 1 in a million effect. Phase 4 is the only place you're going to learn that. And like, it's pretty accurate. The Janssen COVID vaccine, it was the vaccine-induced thrombocytopenia was detected after like 6 cases within the first 6 million doses. So yeah, it does pretty good. Meister Murphy says, do you think if we solve cancer, we'll solve aging? I guess they're two similar problems. But I do think they're separate. Like, you're gonna keep aging even if we figure out how to, like, cure any cancer that pops up in your old age. But so yeah, they're separate problems. The leftist mafia are going to invite you. Oh, cool. Yeah, I definitely go on. I definitely talk to them. Yeah. Awesome. Tim Farrell says, please do this again. I absolutely will. Matt T says, I keep encountering people who claim mRNA is CRISPR. Can you explain the difference? It seems obvious. Absolutely. So let me just see if I can find a helpful graphic of CRISPR. So CRISPR involves multiple components, and an mRNA vaccine does not contain those components. So the important thing with CRISPR is it has to involve an enzyme called Cas. Usually it's Cas9. What this enzyme does, is this a clear picture? We'll just go with this. What this Cas9 enzyme does is it holds this guide RNA, which you design as a researcher or someone designing a drug. And this guide RNA will be bound by the enzyme, and the enzyme will use it to seek out a specific sequence. And once it finds that specific sequence in DNA, then and only then will the enzyme cleave the DNA, allowing for insertion of your own piece of DNA that you want the cell to integrate into its genome. That's one use for CRISPR. It can also just cleave, and if you want it to just cleave and serve whatever purpose you want there, then it can do that too. But yeah, mRNA vaccines don't have Cas9. It doesn't have a guide RNA, which is a specific piece of RNA. Yeah, it doesn't have any of those components, so it's very, very different. Are there... Julian Tran asks, are there any preventative measures that we can take to reduce any inflammation effects from infection? Yeah, I mean, there are drugs that, specifically for COVID, to treat the inflammatory phase, like tocilizumab and baricitinib and corticosteroids. Excuse me. If you're asking generally for infections, I don't know. I mean, talk to your doctor about that if you want, if you have a specific case. But I mean, other than like NSAIDs, you know, non-steroidal anti-inflammatories, I don't really know if there are specific things you can do for like other infections. All right, try to get to the end of this. Ultimate Weapon 2000 says, not sure if you research after your video on Riza Islam, but he's part of the NOI that damaged my family relationship. I'm sorry to hear that. Yeah, I know some things about Riza Islam, but not too much about like who he is personally. I just know that he spreads garbage misinformation. Excuse me, I'm talking too much. David Palermo asked, Campbell is obsessed with excess deaths. Do you have anything to share different from your very good video you did a while ago? Susan Oliver's channel. She's on top of covering John Campbell's nonsense. Look to her if you want to see debunks of John Campbell. Dan asks, do you consider it impossible to have side effects in a future 5 to 10 years may be caused by the vaccine? How someone be sure about it? Yeah, it's not considered a possibility. The reason being, when you have a side effect from vaccines, I'm looking for this CHOP article that talks about it. It usually comes up when I search for it. Why isn't it coming up? Anyway, while I look for it, the side effects that you get from vaccines, again, unless it's anaphylaxis from a specific ingredient, it's going to be because your immune system's responding to the antigen and you're feeling crummy from your immune response. And sometimes that immune response can cause other things to happen that you don't want. But once that immune response is done, in other words, once your immune response has instilled memory cells in you, you have an immune memory, then there's really nothing conceivable to cause a new adverse event. And that timeline is like 6 to 8 weeks. So here's a nice article from CHOP, the Children's Hospital of Philadelphia, on that topic. The pertinent paragraph is, let's see if I can. Yeah. So like it basically describes like the history of vaccine side effects and how pretty much every known vaccine side effect occurs within like the first 8 weeks of when the dose was given. Okay. I think we're to the end. I'll take these last few questions and then I do have to go to bed. We're done after this, but I appreciate you all being here. This is always really fun for me, so we'll definitely do it again. But Dominion asks, how did you decide you want to pursue a PhD? I've thought about doing one, but it seems like you should be pretty certain you want to dedicate your life to something. It's a funny story actually. So I said I want to finish, but okay, I'll tell the story and then I'm done. So like I knew I wanted to be a scientist, but I didn't know what kind of scientist I wanted to be. And then I had a really awesome AP biology teacher when I was in high school, and that was when I kind of decided I want to go to school to learn about cell and molecular biology. Excuse me. Then I got to my undergrad and I had a fantastic professor who I still keep in touch with from time to time today, who I did undergraduate work in his lab. I went to a small university, so I got to have like a lot of like hands-on and one-on-one time with other students and professors. And there was at one point where he said, you know, I think that you're really good at this and you should go get your PhD. And I was like, what does that mean? Like, what does that look like? I don't know what getting a PhD looks like. But I still said, okay, that sounds good. So that's pretty much what happened. I like did some summer internships to get more experience in research, decided I like research and I want to do it in grad school. So then I went to grad school and got my PhD, and yeah, I think that like my path going right from undergrad to graduate school, I knew a lot of people who like took years off and worked like lab jobs before they went to their PhD. I think that's a pretty good plan, honestly. Just like make sure that that's what you want to do and you're willing to accept those like 4 to 6 years of making very little money doing your PhD. So yeah, that's my story in a nutshell. But yeah, basically because I did a lot of lab work as an undergrad and internships, I knew that I wanted to do a PhD where I was going to do a lot of lab work. But yeah. Anyway, go back and do a PhD if you want, or not go back, pursue one if you want. I encourage it, but yeah, make sure that you that it's really what you want, and ask the grad students to tell you the horror stories. Don't be shy to get them to tell you the horror stories, because that is important. You need to know the worst of what you're getting into so that you can avoid it and not make the same mistakes they did. Anyway, that's all for tonight. I hope you all enjoyed this Q&A. I sure did. And hopefully I'll have a new recorded video next week, but if not, it'll come maybe the week after. But thanks everyone. Thank you for the super chats. Really appreciate y'all being here, and I'll see you next time. Bye-bye.